All right, so it's a real pleasure to be here in Vancouver, and I want to thank Simon for hosting me today. We went to a surrealist exhibition, so you may find this lecture to be somewhat surreal. So I'm going to do a couple things in the context of about 50 minutes. I'm going to talk mainly about the issue that drug addiction is a brain disease, and I'm going to try and explain to you what the mechanism for that is as well as we know it to date. And I'm going to focus on the common elements to addiction, but I'm going to use two drugs in the process. I'll talk about cocaine and I'll talk about alcohol. But before I do that, I'm going to also give you a little rudimentary review of neuropharmacology and the neurobiology of stress. All right? So what is addiction? Well, from the brain perspective, addiction is a reward deficit disorder. It's a stimulus response perseveration disorder. It's a stress surfeit disorder. That's the opposite of a deficit. We had that during the Clinton administration in the United States. That's the last time when they doubled the NIH budget. We had a budget surfeit. Right? And it's a self-regulation disorder, and I don't have to tell you all that. All right, but one of the things that all of the work in addiction is predicated on is to understand how the brain works. And in understanding how the brain works, you need to remember how chemical neurotransmission works. So I'm going to start with the sublime and move to the ridiculous in the course of this lecture. So we have cells in our body, you know that. Different cells do different things. Neurons are special cells because they're the only cells that talk to each other. Now, when you're high on drugs, sometimes you think your skin cells are talking to each other, but they're not. Right? So the only cells that really talk to each other are neurons or nerve cells. And they do that because they have processes and two forms of transmission. They use electrical signals, and they also use chemical signals. So the three parts of a neuron, which are shown here in a 100-fold blow-up of your real brain cells, are the nucleolus and nucleus, where your genetic material is and the machinery of the cell is. And then you have these fibers that come out that are called axons, that are the sending fibers, and then you have the receiving end, right? Which is called the dendrite. So you can schematize this and blow it up here. And basically, I've used dopamine as an example because we'll talk about dopamine. It has a prominent role in the reinforcing effects of cocaine. But you can see here that the nerve cell is now blown up, and you can see the dendrites, which look like the horns of your elk, and the cell body and the nucleus, and inside that is your genetic material, and then the axon that goes out and the terminals. But there's something unique about our brains that are different from, your iPhone. Your iPhone has wires in it, and they're connected from A to B, and they're different colors. We have that in our brains with neurons. They're different colors, and they're connected from A to B in different ways. But what we have that your iPhone doesn't have is when the electrical signal gets to that terminal area, which is blown up here, you end up releasing chemicals. And those chemicals are the whole basis of addiction, of stress, and all pharmacotherapeutic drugs that are used in the treatment of mental illness. And so in this example, when an electrical signal gets down to that terminal large enough to release dopamine, then these little packets of dopamine move to the surface and they're released into the intracellular space, and they go to the other side to another cell, which is a dendrite of another cell, and they bind to a receptor, which are these little things here. How much dopamine is in your synapse is actually one of the neuropharmacological things we know a lot about. And cocaine, methamphetamine, D-amphetamine, have this enormous effect of increasing the amount of dopamine in that synapse. All right? That's one of their main neuropharmacological mechanisms of action. And they do it in two ways. They do it by enhancing the release. They move the packets to the surface faster. That's what methamphetamine does. But cocaine blocks very strongly the reuptake of dopamine because you have to get rid of it once it gets in the synapse. So it's taken back up. When it's taken back up, it then gets repackaged and resynthesized. but what cocaine does is blocks that reuptake. So cocaine increases dopamine in the synapse, and methamphetamine increases dopamine in the synapse. And other drugs of abuse also do that, although they do it indirectly, as we'll see in a few minutes. All right, now, that's your lesson in neuropharmacology. I'm sorry, I'm not going to go into any further detail on that, but keep it in the back of your mind. Now, what about stress? What is stress? Well, everybody has their own definition of stress. These are two classic definitions. Non-specific, common result of any demand upon the body. That's Hans Selye from Canada, who defined that many, many years ago. And then a more modern definition, stress is anything which causes an alteration of psychological homeostatic processes. You know what homeostasis is? That's where you keep everything in the normal range. Most of us have blood pressure around 70 to 120. We have body temperatures at 98.6. And we have emotions that fluctuate, but they stay in the homeostatic range, right? Stress is when you're pushed out of that range. So the next slide is a quote from a very popular writer who just passed to the other side, Kurt Vonnegut. And he wrote this in 1950, and I don't know where he got his information, because I don't think Kurt Vonnegut went to medical school. But in fact, he got it all right. And I was at the Salk Institute working with Wiley Vale and Catherine Riviere and Jean Riviere and Joachim Spies when the chemicals that Kurt Vonnegut were talking about were actually discovered in the brain. So bear with me, we're going to go through a quote. It'll take a few minutes, but it tells a really nice story. So I saw Kazak out of the corner of my right eye. His eyes were pinwheels, his teeth were white daggers, his slobber was cyanide, his blood was nitroglycerin. He was floating toward me like a zeppelin, hanging lazily in the air. My eyes told my mind about him. My mind sent a message to my hypothalamus, told it to release that's in the base of your brain, your hypothalamus, told it to release the hormone CRF into the short vessels connecting my hypothalamus and my pituitary gland. Your pituitary gland is your master gland, the base of the brain. The CRF inspired my pituitary gland to dump the hormone ACTH into my bloodstream. My pituitary had been making and storing ACTH for just such an occasion. And near the zeppelin came, and some of the ACTH in my bloodstream reached the outer shell of my adrenal gland, which had been making and storing glucocorticoids for emergencies. My adrenal gland added the glucocorticoids to my bloodstream. They went all over my body, changing glycogen into glucose. Glucose was muscle food. It would help me fight like a wildcat or run like a deer. And nearer the zeppelin came. My adrenal gland gave me a shot of adrenaline too. I turned purple as my blood pressure skyrocketed. The adrenaline made my heart go like a burglar alarm. It also stood my hair on end. It also caused coagulants to pour into my bloodstream, so in case I was wounded, my vital juices wouldn't drain away. Everything my body had done so far fell within the normal operating procedures for a human machine. But my body took one defensive measure which I am told was without precedent in medical history. It may have happened because some wire short-circuited or some gasket blew. At any rate, I also retracted my testicles into my abdominal cavity, pulled them up into my fuselage like the landing gear of an airplane, and now they tell me only surgery will bring them down again. So that's how you know this is a quote from Kurt Vonnegut, okay? But he must have been in a lab somewhere, because if you have rats, like we do in my laboratory, and you have them old rats, we call them retired breeders. I think I probably am rapidly approaching that category. The retired breeders are huge. They can be almost a kilogram in weight, and they have huge testicles which drag on the ground. And if they're stressed, they do retract them into their abdominal cavity. But you don't need surgery to bring them back down. All you need is a cute female rat walking past their cage. Okay, so what Vonnegut was talking about, and what I described in that quote, which is amusing but true, everything in it is true except for the testicles, is in fact a neuropeptide that was discovered in 1980 by Wiley Vale and collaborators at the Salk Institute. And I didn't do any of that discovery, but I was the first person to inject this peptide into the brain of a rat and observe the behavior. And so CRF, or corticotropin releasing factor, does inspire your pituitary to release ACTH and glucocorticoids ultimately from your adrenal gland. And the part that I worked on, and we still work on, is we found that CRF in the amygdala, which is part of our temporal lobe, actually mediates behavioral responses to stressors.